So at this temperature, because spores cannot be killed, so it means we cannot achieve a complete sterilization. When I say sterilization, what do you mean by sterilization?
For making something free from bacteria or
Yes,
living microorganisms.
yes, 100%, 100% free from microbes, 100% free from any living thing. This process is known as sterilization. That is why, you know, when we perform surgery, all the equipment, all the instruments should be sterilized, should be sterilized. Everything should be sterilized, because if any instrument is not sterilized, then the chances of bacterial transmission, viral transmission, or any microbial transmission will be there. So if we boil the instrument, if we boil the instrument, sterilization cannot be achieved completely. By if bacterial spores are present on the instrument, so those spores cannot be killed even in 100-degree. So that is why we have another technique. We will discuss the sterilization techniques later, maybe at the end of or the last classes of microbiology. That is autoclave. Autoclave. Autoclave is actually a specialized, not a machine, it's an equipment, a specialized equipment in which we can achieve a temperature, of 121 degrees centigrade. Under high pressure, autoclave, it has a temperature of 121 degree for 15 minutes. So this is one of the type of sterilization method which can cause 100% free from microbe. So all the instruments, surgical instruments, surgical gauzes, everything which we use in like in medicine, in surgery, those equipment, those instruments should have to be autoclave. So autoclave is a procedure in which we place all the things, maybe like the things which we want to sterilize, in a autoclave, and in the autoclave the temperature, it goes up to 121 degrees centigrade for 15 minutes, and after 15 minutes all the spores will be killed as well. So all the microbes, spores, everything will be killed at this temperature and within this time, 15 minutes. So this is one of the process of sterilization. So that is why the spores of bacteria can be killed only through this autoclave method. Otherwise, the bacterial spore, they are resistant against harsh condition. So this is actually a significance of these spores. So let's suppose I give you a clinical case. Let's discuss. Let's suppose you have a you and your friend, you both are going you went outside for a picnic, and while walking on a road, your friend, he got some injuries. So which you can say treatment it will comes in your mind? Which treatment it will comes in your mind if you get some road injuries or any kind of this road traffic accident? So which
Tetanus का
kind of
shot, sir.
Vaccine for tetanus,
very good,
maybe.
anti-tetanus toxide. That is not vaccine, it is anti-tetanus toxide. that is known as TT. Generally, we call it TT injection. Okay, that is tetanus toxoid. So why we do this? Because the spores of Clostridium tetani, this is a bacteria which cause tetanus, Clostridium tetani. It is present in a spore form in our soil, on a road. So if somebody got some injury, road traffic accident, we give a anti-tetanus toxoid, so that the bacteria, if this Clostridium spore enter into the body and produce any toxin, so that our body can neutralize those toxins. So this is a significance, clinical significance of these spores. So this is that was one example of this spore. So Clostridium tetani, its spores are present in soil. So remember, whenever you get any injury, you should need to get a dose of tetanus toxoid, anti-tetanus toxoid or TT injection. Understood, class?
Yes, sir.
Okay. So okay, I have explained you about this. So these are the different types of spores you can see within the bacterial cell. Okay, so different species, they have different shape, morphology of spore, as well as different site of spores which are present within the cell. So next we have gram staining technique, that we will discuss later in our maybe next class. We will first discuss about the staining techniques, how we stain the bacteria, and how we classify the bacteria according to the gram staining. Gram negative, which bacteria are gram negative, which bacteria are gram positive. The important thing is here, the nomenclature. I think this is, you must already know about it. When we name the bacteria, we actually use genus and species. So all the bacteria, when we write their name, we write their genus and their species. So some bacteria, they also have a common name. We don't use any species name or genus name. We just use their common name or informal name. Like meningococcus, meningococci, as the name suggests, meningococcus. So this bacteria cause meningitis. So that's why we call it meningococci. But actually, the formal name of this bacteria is Neisseria meningitis. The actual formal name is Neisseria meningitis, but informal name, commonly we call it meningococcal or meningococci. Okay, gonococcus, that this is a bacteria which cause gonorrhea. Gonorrhea is a sexually transmitted disease. So this is an informal name. So some bacteria we use a informal name, some bacteria, mostly bacteria we use scientific name, like we use genus and their species. Okay, so any question till now?
No questions.